Hi, I'm Trent Blizzard, and I am pleased to introduce today Bryn Dentinger, who is going to be speaking to us about the phylogenomics to resolve the evolutionary radiation of Boletaceae. I don't know if I said any of that right, Bryn. I'm gonna pass this over to you here in just a second. Bryn is out of the University of Utah in Salt Lake, where he works and operates in the Dentinger lab. And he kind of came into this as a forager and is very involved in the world of Boletus and Boletus edulis, and I think is one of the world's experts in this topic. So I'm excited to turn it over, and I'm going to sit back and listen to you for an hour, and I'm going to tell everybody else there's a chat. Hop on the chat and say hi. It's open to everybody. And there's also a Q&A. If you put your questions in the Q&A, Bryn's going to try to monitor those and answer them during the class. So feel free to hop in the chat and communicate during the session. Although I don't think you'll be watching the chat. That's probably too much. But you will be watching the Q&A. So I think we're ready to go if you are.
All right, thank you, Trent. I just saw somebody in the chat. Yeah, there's no, I don't hear anybody, so that's great. I'll let you know if you end up unmuting by accident. I will try to monitor the Q&A during the presentation. So if you have questions in the middle of my presentation, feel free to add something, and I'll be checking that regularly. And if we end up running out of time, I'll be able to maybe answer questions at the end, and then if not there, perhaps through email or some other means. So let me share my screen. And hopefully you all can see that. Now I got to get rid of some stuff here. Okay. Oh, I'll hide that too.
Bryn, that looks good. So
Okay.
we can see that.
Great. So thank you all for being here tonight. It's really an honor to be able to talk to the NAMA community about this work that I've been doing really since my PhD, when I began my PhD way back in the early 2000s. So this is kind of a labor of love, and it obviously takes a long time to get to where I've gotten, and it's just really an honor to be able to share it with everybody. So with that, Trent did explain, or did pronounce all the words of my title correctly, and hopefully I'll be able to explain what some of these things mean if you aren't familiar with these concepts. And if I don't explain it well, you will hopefully let me know. Okay, now I'm having trouble advancing the slide. There we go. Okay, so one of the things that's always intrigued me about the bolete family, which is the Boletaceae, is illustrated here with Boletus edulis, and many people that are watching this are likely familiar with the variation that you find in Boletus edulis. So we have color forms, we have different forms of stipe, heights and thicknesses, different potential hosts, and they're geographically sometimes sort of concentrated, these forms. But at the same time, the boletes display a high degree of convergence. So you can look at Boletus variopes from the Porcini group next to Tylopolis phelleus, and at a very coarse level, they look very similar. And a lot of people who don't have experience looking at mushrooms would think that they're the same thing before they taste them, of course. But this has always fascinated me, this sort of, on the one hand, this like high degree of variation, and on the other hand, this high degree of convergence. And it's always been kind of a mystery and baffled me. And so that's sort of the motivation behind a lot of this work, is trying to understand that variation, how it came about over evolutionary time, and what the mechanisms are that drove it. Okay, so starting with how Boletaceae used to be classified, there was a large emphasis on morphology, and this is true for most mushrooms, all mushrooms really. And some of the features that were really important in the classification of the Boletaceae included spores, of course, features of the hymenophore, that is the spore-bearing structure on the underside of the cap, both anatomically, how they're arranged, as well as microscopically, as illustrated here, what they look like. So if you were to slice through like a tube, you might see this sort of pattern of the cells, or you might see this sort of pattern of the cells. And these were features that were used for a long time to classify the Boletaceae. Here are some examples of basidiospores that were emphasized in the 80s by Pigler and Young. This is the trauma that I was just discussing, where if you slice through a tube, you might see these different arrangements of the cells. And then specialized cells, whether they're present and what they look like cystidia, were also important and still important for identification. And then finally, there's also some secondary metabolite chemistry. These are pigments in this case. As many of you are probably aware, a lot of the Boletaceae have really bright pigments as well as color changes. So when you cut open one of these mushrooms, you might see different colors forming, like blue or purple or brown, black, those kinds of things. And so these are based on this chemistry here that's being illustrated on the lower right. So these are the features that used to be used to classify them. But a lot of mushrooms these days now are classified primarily using molecular methods, using DNA sequences. And that's because of this problem that I mentioned early on about the high degree of variation on the one hand and convergence on the other. Those two in combination make morphology a really misleading set of characters to use when you want to try to figure out what things are related to one another. So the DNA-based approach has really taken over the field of mycology systematics, that is the study of how things are related, since maybe the late 90s or so. And the Boletaceae have always been of interest to a number of people because they've really been a difficult group to figure out, not just morphologically, but also it turns out DNA sequencing has not really given us a whole lot of information about how things are related. So one of the early studies was this study in 2001 that identified the Boletaceae as a radiation. And what's meant by that is there was a lot of species that formed in a very short period of time. So that's called a radiation. You can think a really, I think, apt comparison is the songbirds. So the songbirds are really diverse and they sort of exploded on the scene in a very short period of time. So that's sort of the same thing we're dealing with the porcini family Boletaceae. Things got a little more and more, got the trees of Boletaceae got bigger and bigger, so people were sampling more and more of them and added more genes, and they got more colorful, but ultimately the problem remained that the backbone, that is the relationships amongst all these branches that you see on the tree, has never received statistical support. So we've never been able to resolve how they are all related to one another. And so that's been one of the driving motivations behind the work that I'm just about to present. So one of the problems of these radiations is because things diverged really rapidly, they often don't leave any sort of record of their relatedness. It just doesn't get recorded because it takes time for that to happen. And so that's been one of the major problems that we've faced with trying to resolve this radiation. And that can lead to what's known as this incomplete lineage sorting, which I'm not going to go into. But basically, you can imagine that you have signatures of some species that are left in descendant species that are not really showing the true relationship between the two species. I'll just leave it at that. If you have questions later, I can try to explain it better. And the result from a taxonomic perspective is that the boundaries between these different groups have been undefined, and um, that leads to kind of this quagmire, if you will, of when people find new species of Boletaceae and they try to place them in the phylogeny to figure out what genus they belong to. It turns out that many times that's not possible. So if you're collecting species from some underdocumented region like the Amazon basin in Guyana or the Congo basin, where there's just been very little survey work, when you try to place them and understand how they're related to the other boletes, they don't show a strong relationship with anything. And so that has led people to um, name new genera for all of these species that they can't place with existing genera. And that has resulted in an exponential increase in the number of genera over the last decade. Um, and it's gotten really out of hand, I would say, because um, we really don't know whether these represent real genera in the sense of how we might think of them morphologically or or be able to identify them in the field. So they're sort it's just been kind of well, for me, I guess I should speak for myself. I can't really keep up with the names, and I find it totally overwhelming to try to identify genera in the field these days because um, you have to know the species before you know the genus, and that doesn't make a whole lot of sense to me. Anyway, the solution to this is more data. So one of the ways you can get around this problem of very low levels of recorded relatedness is by just adding more data, because you know, the more you add, the more likely you are to find something that tells you something about the relationships. The other problem that's been present in prior studies is that there's been a what we call in the field an ascertainment bias, and that is people have not used representative sampling to build their trees. That is, they've left out entire regions of the world, and then also taxa, say rare taxa, that are known to exist. Those haven't been included. And that can really be a problem because you might find records of relationships in those taxa or regions of the world. And if you're missing them, obviously you're not going to be able to figure that out. So that's another possible solution that needs to be explored. And then finally, there's a technical aspect of this, which is you can use statistical methods to account for this incomplete lineage sorting, and that's called coalescence. Okay. So, interestingly, there's another problem here, which is some of the genes that people have used have been positively misleading in regards to the relationships. So I'm showing you, this is a study I published in 2010, where I used 3 different loci, and you can see that the 2 trees on the left and the center do not resolve porcini, Boletus sensostricta, what everyone would recognize as porcini mushrooms, as a monophyletic group. You can see that they are scattered across the tree. And that led, or it contributed to separating Boletus separans, a North American species, from Boletus and placed in the genus Xanthoconium. But a different gene, which is the right-handmost gene tree, actually resolved them as a monophyletic group. And so this is an example of so that meaning that they are all derived from a single common ancestor. They have one parent rather than multiple parents. And this is what we expected from morphology, because porcini are kind of distinctive-ish morphologically within the boletes. And so this indicated that perhaps the problem lies largely in the information that's available with the various genes. And so these genes, although they've been used previously, they have information that is either not useful or is actually giving us the wrong information. Okay. So, what we did to set out on our big project was the rationale for this is that we can generate whole genomes from fungi in a really efficient and relatively cheap way. And that's because the genome size of fungi and mushrooms is small relative to other groups of organisms like plants and animals. So you can see this chart is showing you that this is the range. This is a bit out of date, but this is the range of genome sizes of fungi. Um, and they are, magnitudes smaller than flowering plants or mammals. And so that makes it really kind of easy for us to generate genomic data. So then we also took another approach to fill in or to try to minimize this ascertainment bias problem, and we sought to include specimens from as many regions of the world as we possibly could, especially from regions that have not been sampled or are underrepresented in prior studies. And so this is a map of where our material came from, and you can see we've covered all of the continents that the porcini family is known to occur on. It's of course not completely covered because that's practically impossible, but you can see we've gotten material from places that are severely undersurveyed for fungi, like Madagascar or the Congo Basin of Africa, um, Australia, and the Indonesian islands in Southeast Asia, these kinds of places. Okay. And so then what we did is we took and we're up to, I don't know actually know how many now, but for the study that I'm gonna talk to you about today, we had over 400 specimens collected, and this is a map of where those specimens came from. So you can see there's still a bias in terms of the numbers. There's a bias towards North America, and I'll explain why there's this kind of massive bias here in a second. You'll see. As well as Southeast Asia has a high number, but you can see we really included a lot from Africa, and this is due to some recent fieldwork that I have done in collaboration with Terry Hinkle and Kathy Aim in Cameroon. So this is the first time really that African taxa have been included in a substantial way in a Boletaceae phylogenetic study, excuse me. And so then our approach was we used this sequencing method called shotgun sequencing. And basically what you do is you take the genome from the mushroom, you break it up into small pieces, and then you sequence every one of those pieces. And then you have to stitch it back together. And that's what the assembly and ortholog extraction part is. So we stitch it back together, and then we take portions of the genome that are the same between the different species so we can build what is known as a sequence alignment. We then infer the phylogenies from those alignments, and that's kind of the basic approach. So this is the result of many years of work, both field work and recent phylogenomic work. But also, I want to emphasize it's not just my effort here. A lot of the effort that's represented in this phylogeny is by past collectors and also some present collectors. Maybe some of you that are watching this have contributed to this project. Thank you very much. But we relied heavily on specimens that already exist in Fungaria worldwide. And that's, simply due to logistics. It's impossible for us to go to all these places and be there at the right time to collect all this stuff ourselves. That's kind of a ridiculous idea. So we really had to rely heavily on existing collections to build this. Now I'm going to zoom in on this, so don't worry if you're squinting at your screen. Don't worry, I'm going to take you through these. The colors represent the subfamilies of the Boletaceae. So I'm going to go through each of those subfamilies separately now. So the first subfamily, which is in green, is the subfamily Boletoideae, and that is the subfamily that includes porcini in the strict sense, Boletus sensu stricto. I just realized I can't see the Q&A. What do I have to do here?
You want me to read you the question, Bryn?
I got it. I had just hidden my control panel. Okay. Yeah, before you can, oh yeah, so sorry. Sorry, I should have looked at this previously. The question was, how do you mean that you need to know the species before you can determine the genus? What I mean in this case is there are genera out there that are not, you cannot distinguish them from other genera without knowing what the species is. And a lot of these new genera that have been described in the last 5-10 years only have one species in them, or maybe just a couple of species in them. So this is what I mean, like to know the genus, you have to know the species. And that's, from my perspective, that's not a practical use of the generic rank in taxonomy. From my perspective, genera should be something you can recognize in the field, at least in as many cases as possible, that are collections of species. So that's what I mean by that. Hopefully I answered that question well enough. You can follow up later if I didn't. Okay. All right. So within the Boletoideae, these are the genera that are currently placed in this subfamily. So this group up here is composed of these genera: Strobilomyces, Porphyrillis, Xanthoconium sensu stricto, that is Xanthoconium based on the type, which is Straminium. That's where Straminium occurs, is up here, not with separans, which is down here. And then a recently described genus, Neotropicomus. And there's a few other things, and if you can see well enough, you might also notice some other names in here, unidentified things that are named Tylopolis, Xericomus, and that is because these are field IDs, and they're collected in places where there's very little taxonomic literature available, and so it remains to be seen what genera they belong to. I'll zoom in on Boletus in the next slide, so I won't cover this at the moment. And then down here, we have the genera Tylopolis sensu stricto, that is Tylopolis, that are all related to the type of this genus Tylopolis phellius. They all belong in this group. Imlaria, Xericomelus, and Hortiboletus. So those are the groups that fall into this section of Boletoideae. Okay. Now zooming in on Boletus sensu stricto or Porcini sensu stricto, this is what the genomic tree looks like. Of course, this is not exhaustive for all the species known in the group. We do have data sets that are as comprehensive as possible using publicly available data, as well as a lot of unpublished data that we have from around the world. And I'll just point out one thing here, which is the earliest, so the first group that splits off is down here, and it's represented by this species that I helped to describe from Thailand, Albu brunescens, but also a species, a new species that currently does not have a name, from Texas, and I believe also along the Gulf Coast. And this is a citizen scientist collection. And so this, I think, is a good illustration of just how important citizen science is to diversity studies in the fungi, because we can't all be everywhere all at once where we need to be able to collect these things opportunistically. So the more people out there, the more we can discover these new things. So that's been a really important aspect of some of this work. Okay, then moving down the tree is these two subfamilies, Swilliloideae, and that's composed of this long list of genera on the right, as you can see, typified by Swillellus. And then the next subfamily, which shares a parent with Swilloloidy is the Ostroboletoidy, and you can see it's much smaller in our sampling. That is also reflective of the species diversity that is known for this group. And that is typified by Ostrobolides, but also the genera Fistulinella, Veloporphyrilis, Bothia, and Coeliocasis. Coeliocasis, if you're not familiar, is a sequestrate that is a truffle-like bolete from Australia. Moving down, this purple group is the Lexanoidy, and it's typified by Lexinum. And if you recall from the map, which you can sort of see here, that really large number in North America, 181, that's inflated because of all of the sampling we did of the Lexinum. And the reason we did so much sampling in Lexinum is we had the opportunity to sequence the types from the Smith collections in Michigan. And Alex Smith did a lot of work in the taxonomy of Lexinum. And so most of these are types. Yeah, nearly all of these are going to be types, these MISH samples of Lexinum. So we are not focusing on the taxonomy of Lexinum. We're collaborating with people that are, but that's partly why we have such a large number in North America, because most of these are from North America. But this is not a strictly North American clade. So Spongispora is a recently described genus from Southeast Asia. Spongiforma is a sequestrate bolete from Southeast Asia. Pseudo-Austrobolides comes from Australia. Redibolidis is known from Southeast Asia, temperate East Asia, and North America. And Binderobolides, which is from Guyana. So it's quite geographically diverse. And that's true for all of these subfamilies. Moving down to the bottom groups, there's two subfamilies here. The one on top is the Xerocomoideae, and that's because this is where Xerocomus, in the strict sense, occurs. That is all the species that are related to Xerocomus subtomentosus, which is the type of Xerocomus. But also includes Phyloporus, Tubocida, which is an African endemic genus. It looks exactly like you would think it looks, a Xerocomus-like thing, but it's got these really cool cystidia. Boletellus, Hemioporus, Velobolidis, which is a new monotypic genus from Australia, Hemilexinum, Aureobolidis, and Butyribolidis. These all belong in the Xerocomoideae. And then next to it is this subfamily Zangioideae. And this has currently only 6 genera represented in it. And it's I find it kind of funny because all of those genera except one are named after people. Zangia, Chuya, Royungia, Wakefieldia, Haria, those are all named after people. Kind of a fun fact. Okay, and then at the very base of the tree, so the oldest groups of Boletaceae are these two groups. And we named this in this study, which was just published, the Phylobolatelloideae, and that's because this really unusual, rare genus Phyloboletellus, we were able to sequence the type from Michigan, and it came out at the base of everything up to this point that I've discussed. We also do know from another study that's being published, but also from our own internal data sets, that Pseudobolidis also belongs down here in the Phylobolatelloideae. So the Phylobolatelloideae is not a very diverse group. It's quite old and currently consists only of Phyloboletellus and Pseudobolidis. And then finally, we've known for a long time that the earliest group that represents the Boletaceae, that is the oldest group that represents the Boletaceae, is composed of Chalciporus, as well as the genus Buchwaldobolidis. And these are interesting ecologically because they show patterns of saprotrophic capacity, or even in the case of Buchwaldobolidis, mycoparasitic capacity. That is, it lives on other fungi. And that's quite different than the ecology that we know of for most of the other Boletaceae, which are all ectomycorrhizal, meaning they form associations with the roots of certain trees and other plants. So that's the overview of this giant study. It's published in Mycologia just recently. If anybody wants a copy of the manuscript, just maybe I'll just send it to Trent and he can pass it around. I think it's also available on ResearchGate or somewhere else. Okay. Let me see how I'm doing on time. Yeah, good. So should I pause and maybe take a question or two? That was probably information overload.
I'm not, this is Trent, I'm not actually able to ask a question in here, like the other attendees, so I was just gonna verbalize mine.
Sure.
This concept of using phylogenomics, has this been happening in other genuses around kingdom fungi?
Yeah, so the genomic approach to building phylogenies is taking over now. And that's for the reasons I've explained previously about a lot of the poor statistical support for relationships in the past seems to be no longer an issue in many cases when we use whole genome sequences. Did that answer your question, Trent?
Yes.
Cool. I'm looking at the chat. Looks like there is somebody asked about gyroporus. Yeah, so gyroporus does not belong to the Boletaceae. That genus actually belongs to a different group within the order Boletales, but is not related to is not very closely related to porcini and these other genera that I've mentioned. And then there's in the Q&A, yeah, other mushroom clades that exhibit high levels of evolutionary radiation or ILS. Yes, likely. And some of these are going to be like Russelaceae, as well as, let me think, Cortinariaceae. And, but the difference between those is, well, except for Russelaceae, which I can't remember now how old it is. The difference, at least with the Cortinariaceae, is it's a very young family. And so that radiation has very different features in it than the one that we're discussing. One thing I didn't mention was just how old the Boletaceae are. And they're very old. We dated the origin to around 100 million years. But the Cortinariaceae is going to be, much, much younger, maybe 10 to 20 million years. Okay. And then do you think the radiation happened when boletes figured out how to live mycorrhizal lifestyles? Yeah. Or rather, why is it that the saprophytic parasitic ones were so close to the base? Yeah. So Walker, that's a great question and you've sort of hit the nail on the head. That is the current thinking, is that that's exactly what happened, is the acquisition of the mycorrhizal habit led to this explosion of diversity. So that, yeah, other studies have shown that. So yeah, cool. Gyroporus. Gyroporus is mycorrhizal, and it is in the Boletales, but it's in a different family of the Boletales. It's related to scleroderma. Okay. I will continue on then. Why can't I see? Oh, there we go. Just changing my Zoom settings here. Oh, it disappeared again. All right. So I'm going to transition now into looking at a very fine scale, which is looking at how Boletus edulis, that species, is becoming multiple species. So as a reminder, porcini sensu stricto is just one group of the Boletaceae. This is a species level phylogeny from some older renditions of my internal data set that is not yet published. And you'll see up at the top here is Boletus edulis, and it is this complex of morphospecies. And many of you may be familiar with some of the names that have been given to these morphotypes. So this is Boletus edulis var clavipes. There's Boletus chippewanensis. This would be the Boletus edulis var betulicula. This was segregated as a species. This was called Boletus rubriceps. And then this is a sort of typical form in Europe where you get this dark bronze colored cap. But then these were all collected by me at one location in one day. And you can see that this variation in color and stipe, really, it doesn't, there's not very strict boundaries around these different morphotypes. So we wanted to know, we're trying to understand where these variants come from and what they look like genetically. That's one motivation, but also stepping back and looking at Boletus edulis as a species, and maybe these are just color morphs that can occur at random. Looking at what is driving the cohesion or separation of groups within the species. Okay, there's a question. There are other boletes that associate with insects like Boletinellus merulioides. Boletinellus merulioides is a really interesting one. This is called the ash tree bolete, and there's one study that suggested that it is associated with aphids that feed on the roots of ash trees. But it turns out that there's no information beyond just that simple observation. So I would say that we don't really know exactly what the relationship between Boletinellus and aphids is. That's part answer. The other part is there's not any other association known at present between boletes and other insects. Boletes in the sense of like fleshy mushrooms. Okay. I'm supposed to be clicking answer live, I guess, right? Okay. Cool. All right. So just a little overview on Boletus edulis. It is found throughout the northern hemisphere. You can see this map is showing its known distribution. Then its native distribution. It occurs from sea level to subalpine and tundra habitats, and that's one reason that we find it very interesting, because it's got this huge ecological amplitude. And so what is maintaining that ability to grow it in all these different places? And then it also associates with many, many different hosts. In fact, nearly all the native EM host trees in the whole Arctic region are known to be host to Boletus edulis. So it's also got not just its ability to grow at sea level to subalpine habitats, but it's also able to grow with basically every known possible host. So it's really kind of an interesting species in that regard, because there aren't very many examples of this, of species with this level of ecological and host breadth. Okay, it also has been established in non-native regions several times, and these are some examples of where it's been known to establish. And that's really useful for us because it allows us to look at temporal change. So we know, for example, when the trees that are host to them were brought into these regions, and that gives us a chance to look at genetic diversity of the species in those regions over a fixed period of time, because we know exactly how long it's been there. Okay, for those of us, I'm just checking the Q&A. For those of us collecting bullets in North America using standard texts and keys, can you comment at some point on how we should think about your results affecting our field classification exercises, which of course based mostly on morphology and location? How do you begin to reconcile this with the phylogenetic insights? I'm going to answer that at the end, because I'm also going to turn that back on to all of you, because I don't think that it's necessarily up to me how we deal with this stuff. So if you can wait to the end, that would be great. All right. So, the first question you might ask is, do we have one or more species of Boletus edulis? And if you look at the ITS tree, the species, the morphospecies that are also geographically restricted or thought to be geographically restricted, they don't actually sort according to the morphology or the geography in entirety. They sort to some degree, but not completely. And this includes Boletus rubriceps, by the way. So this was our first clue that maybe there's something more interesting going on than just your standard off-the-shelf speciation. And then when we compared this with a different gene, so maybe the ITS is not very good at distinguishing species. So we looked at another gene that's commonly used for determining species, and we get the same result, where there is some degree of geographic clustering or morphological clustering, but it's not complete. So we can get red-capped species occurring in brown-capped clades and white-capped species occurring in red-capped clades. That kind of thing is what's happening. And so this was our initial consideration of Boletus edulis taxonomy. And so we treated this as being a species that is in process of becoming more species, but it's incomplete. So this is a figure from a paper back in the early year 2000s that sort of described the speciation process from going from one species down here to two species up here as being a gradual process where you get these different levels of distinctiveness. And it's not until you get up here where the species are fully separated genetically, and you can detect that using genetic methods. So we're fairly certain that we are dealing with a species in the gray zone here. And that's why some people consider it more than one species, while as others consider it one species. And so, I think it's up to all of us to maybe just agree, if we can, on how we're going to treat this for the time being. And, I'll make my argument, hopefully, that we really should just be calling it one with one name at present. But that's of course going to change in the future if the speciation process continues. But we're talking millions of years, so none of us will be around for that. Okay. So then we wanted more resolution than just these one gene or two gene phylogenies. And so what we did is we generated whole genome sequences from 160 specimens of Boletus edulis, and then identified individual genetic variants across all 160 specimens. That's what this SNP is. So we identified just under 1 million variants across these genomes, and that allows us to look at very fine scale resolution of relatedness and other things, genetic things. So the first thing I'm showing you here is a phylogeny of Boletus edulis using these SNPs. And the phylogeny is colored according to geography. And then this is the map of the geographic locations of those individual specimens. And what you're seeing here are pie charts that represent the genetic content of the different groups. So you can see here, let's just talk about Boletus rubriceps for a second. You can see rubriceps forms a very clear clade with the SNP data. And if you look at all the pie charts, they're almost all of them are nearly 100% rubricep type. And that's true for many of these other groups as well. So at first glance, you would say that this supports dividing Boletus edulis into multiple species that have geographic isolated ranges. There's a couple of problems. One is this here, this light green is Boletus edulis from certain areas along the west coast, and the range overlaps with what's typically known as the west coast Boletus edulis, which includes the var grand edulis. There's zero genetic data to separate grand edulis from all the other edulis on the west coast. This green one, however, which does not correspond to grand edulis, is distinct. And we don't quite know yet exactly what's happening with that group. But if there were morphological differences between these and the rest of the West Coast edulis, one could conceivably segregate that as a distinct species and call it something new. But as far as we can tell, there are no morphological traits that separate this from the rest of the edulis. Okay, so this was the first pass. And then up here, I'm gonna move my little window. Okay, up here, this gives a slightly more nuanced view of the genetic variation. So the colors correspond again to these geographic lineages. But you'll notice that now we have bars that have different colors in them. So here, just looking up at Alaska, you can see that this sample here is like almost all of the Alaska genotype, but it's got a little bit of this Colorado rubriceps genotype in it. And you can see there's a few other examples of that here. So here, this is we call this East Coast, and there's a couple of individuals where there's still majority East Coast, but there's a little bit of DNA coming in from these other geographic groups. So this is starting to muddy the waters a bit. You can see now that there's actually some genetic variation. There's genetic relatedness within individuals of these populations that correspond to all the other populations. Okay, so how do we, what do we make of that? Okay. So this is a kind of complicated figure that I don't want to labor on too much, but what I will tell you is that these D values indicate the level of detected gene flow between these different populations. And in most cases, the detected levels of gene flow are quite high. So there doesn't seem to be a barrier between recombination of these different populations. And that is one of the ways people have distinguished species in the past. It's probably the way that most people think of species is if two individuals can't mate and produce a viable offspring, then they're different species. And so what we're looking at here is all of these individual lineages that are colored and are geographically isolated or geographically restricted. They're all able to form species. They're all able to form viable offspring with each other. That's what we've detected from the genetics. Now, that's just, we call that the biological species concept, and that's just one way of recognizing species. And I'm not advocating here that that's the way we should be recognizing species in this case, but I think it is something to consider. Okay, so this is a new data set. This is taking the old data set, and we've been able to add a lot more data to it, including we've been able to sample with the help of our friend and colleague Roberto Flores in Guatemala, some of the populations of Edulis that represent the southernmost extent of its native range. And doing that allowed us to, one, identify that these Guatemala populations were really different from the rest, but also allowed us to reevaluate how much of this introgression or the gene flow that we've been detecting, how much that actually exists across the individuals. So you can see that with this expanded data set, we're starting to see a bit more introgression, we're detecting higher levels of gene flow than we did previously. And this is not an unexpected result, because if you imagine how many individuals of Boletus edulis there are just in the Rocky Mountains, and we've only sampled 30 of them, this is another example of ascertainment bias. We have a very small representation of the actual population. So we hope that what we have is representative. That is, we've sampled randomly, and so therefore it represents what the actual population looks like. But that may not be the case. And I think here we're showing that as we add more data, we're actually able to see more and more of this gene flow than we did previously. Okay, so this is the question that we don't really have an answer to, or maybe we don't have an answer that's going to satisfy everyone. But our feeling is that we really ought to be treating this as a single species, and then we can that doesn't keep us from saying, oh, this is the Colorado rubriceps form of Boletus edulis. But I think to to be honest about what we understand from the genetics, we really ought to be treating this as a single species at present. In a million years, maybe we'll be treating them as multiple species, but for now, given the snapshot we have of what's present, I think that's probably the most realistic. Okay, so we do have strong geographic differentiation, but we know they're exchanging genes all the time. Okay, so that's our opinion. All right, so I'm gonna transition into something that a lot of people have had interest in the past, and I can do this rather quickly. But one of the persistent issues in systematics and taxonomy is knowing what the correct name is to apply to modern samples. So you go out and you collect a mushroom and you say, oh, that's Boletus variepes. Well, are you saying that because you actually know what Boletus variepes is, or because you think it's the thing that Charles Peck named back in 1904? It's the latter. And the problem is we actually don't really know what the thing was that Charles Peck named in 1904, because he used very terse and um, kind of not very helpful descriptions of these species. And there's no DNA from his concepts of these names to compare with modern material. And so people have just done their best, but really we don't really know how to apply these names. So we set out to sort of, we tried to provide DNA sequence data from all of Charles Peck's species of porcini and some others that we were interested in, to try to nail these names down. And without sort of stealing my own thunder here, we found a lot of problems. So I'm just going to go through this a little bit. Oh, yeah. So one of the oldest specimens that we were able to get DNA out of is this sample from the type of Boletus gigas. This is a species described by Barclay, based on a collection from Sikkim, India, back in 1849. And we were able to get a genome from it. Okay, so really, these samples are very much like ancient DNA. When you read about people sequencing Neanderthal genomes, this is kind of what the DNA looks like. So we have Neanderthal mushroom DNA. And the problem is that most of the DNA is broken into very tiny pieces. And so that's what this chart is showing you. This is a frequency histogram. So it's a number, it's the counts of the sizes of fragments. And the peak here is 58 base pairs, only 58 letters long. So just for context, like a chromosome, a whole chromosome of a fungus is around 1 million. So imagine having a whole chromosome broken into 58 uh, letter pieces and then having to try to stitch that all together. It's a real nightmare. Anyway, this is why past attempts by people have largely failed to get DNA out of these samples. It's because you can't use what we now view as sort of traditional or legacy approaches using PCR, because PCR will fail with these small fragments because we don't have the right primers to be able to design them for fragments of this size. So we have to take a different approach. And luckily, Illumina sequencing, the sequencing I've been talking about up to this point, the so-called shotgun sequencing. Shotgun sequencing is designed to utilize short fragments already. So we can take this and then just do shotgun sequencing and then try to bioinformatically piece it all together after. So one thing I'll show you is that one of the problems with these old specimens is that there's a lot of stuff in them and on them that is not what you want. It's not Boletus edulis. In this case, we had a bunch of insects, we had a bunch of bacteria, we had molds, and a bunch of other things. And so this is one problem that we have to sort through when we're dealing with data like this. So it's a non-trivial issue, I guess, is the point I'm trying to make. This is a technical thing that I'm just going to skip by. So if you're interested in this, you can ask me about it later. This is all unpublished at the moment, but we're working on getting it published perhaps later this year. So a couple of results from this is we were able to fish out the ITS region, which is sort of the DNA barcode. It's your go-to, your first stop when you want to be able to identify a mushroom to species. And so we did that from these genomes. And so the first couple of results here, that the Boletus gigas, in fact, is not a Boletus at all. It belongs to Lexinum, which is actually backed up by morphology. And then Peck's species, Boletus multipunctus, which has previously been referred to porcini, turns out that it is not porcini at all. It belongs in a completely different group, in the Swilloidae. Okay, so we plugged in all of these Peck types into my internal data set and then figured out where they all belonged. So this is a tree of porcini that has 32 species in it. Sorry, 32 species that are represented by sequences from type specimens. So these are really anchoring the names. There's an additional 13 species that are new to science. They have not been described, that don't have types yet, I guess, by definition. There are 4 species that do not even have physical types. And these are the 4 species in Europe. But luckily, they're not very diverse in Europe and they're very easy to identify. So we don't have a problem with those, luckily. And then we were able to put names, the correct names, at least from the concepts of the original authors, onto modern material and fix those now for the first time. And 3 of these names, which have been referred to porcini in the past, have not been used in over 100 years. But it turns out that some of the species that we call by other names today are in fact these species that Peck first named. So I'm going to go through these very quickly. So this is what we currently think represents the diversity of porcini, sorry, the correct names of porcini in North America. So some of these might be a surprise. So if you're in the Midwest or east of the Rocky Mountains, you might be familiar with a species, whoops, of porcini known as Boletus nobilissimus. It turns out that Peck described this originally as Boletus variepes. So we now know what variepes is, and it's nobilissimus. So nobilissimus is now no longer, it's no longer going to be the correct name for those mushrooms. There's a couple of, so this frustulosis one is interesting. So this was a species named recently as Pseudopinophilus, and our data suggests that Peck actually described this originally as Boletus frustulosus. But there's some really odd morphological features of this species that we're trying to confirm using some new samples. So, don't take any of this as being set in stone at the moment, but it's just kind of a preview of what we probably will be publishing soon. This was an interesting one. So Boletus regenius, which was named recently by David Aurora and Simonini. It turns out that Harry Tiers first described this species as Boletus madiae, and we have very good evidence that these are the same species. And so madiae, because it was described first, has priority. So this would be the preferred name under standard nomenclatural rules. Subserial lessons this is perhaps of no surprise to people that are familiar with these two subspecies named by Dick and Snell. But these two subspecies, subserial lessons and Aurantia ruber, really are the species subserial lessons. And this is a distinct species from Edulis. It's not very close in the tree. What people currently think is Boletus variepes. The species that people currently apply the name Boletus variepes to, Peck named as Boletus leptocephalus. So that's also going to be a big name change for people. But this would have priority based on the current nomenclatural rules. There are exceptions made to these things. So as a community, we could all decide that no, we really want to just keep using the name nobilissimus for variepes because everybody knows it as nobilissimus. And we really want to use variepes for leptocephalus because everybody uses it as variepes. And that would be okay. We just have to make a case for it and sanction those names, and it would be fine. But this is how the rules currently would apply in this case. The variety of variepes named by Smith and Tiers called variety Fagicola. This is a species, a distinct species that doesn't currently have a species name. So this is something that needs to have a name that distinguishes it from the other species. And then there's a couple of these other things. So a lot of people still know Boletus separans as Xanthoconium separans, but it is not related to Xanthoconium. It really does belong in Boletus, and so people should be referring to it as Boletus separans. We have not sequenced the type of this yet, but we have it, and it's in progress. And then finally, Boletus gertrudiae, which is kind of an oddball species named by Peck. This is the same species as Boletus nobilis, so this is no longer a name that people should be using. Okay, and then to kind of tie things up, I kind of prefaced this before, which is citizen scientists have contributed a lot of really important collections. And here are species of porcini that do not have names in North America. And these are, nearly all of these are citizen scientist collections. So this is very exciting to me that we can still find new species of what I regard as the most charismatic and important mushroom species in the world, and we can find them in our backyard. So that's like, people might disagree, but I feel like this is like finding new species of elephants in your backyard. Okay. And then here's some from Asia. You can see There's some really spectacular things like this blue and white. This is only known from a single collection from Taiwan. So really cool looking things. And then some of these are things I've collected. And so that is all I'm going to talk about tonight. And I would love to take a few questions, although I don't have a ton of time. So if we don't have time to get to your questions, then I would love to be able to answer them via email or some other format. Okay. For contrast, can you speak about instances in Boletaceae where several species are being combined into one? Are you familiar with the work being done with Lexinum to combine several species? Yeah, so I mean, I don't agree or disagree with that. I think taxonomy ultimately is an opinion, and ideally we all sort of share the same opinion about it, but we don't have to. So I don't know. I take a very practical approach to things. Um, I think in that if I can't recognize the different, well, okay, let me just back up a bit. So I think the combining the Lexinum species that are being proposed into one is sort of similar or analogous to what we're talking about in Boletus edulis, which is, I think, mycologists, both field mycologists and professional mycologists, have not appreciated variation in the past as well as they could have. So I think there's been a lot of quote unquote splitters. Alex Smith is a great example of that, where every single variation that was observed was recognized as a different taxon. But I think there's a role, there's a role for understanding biological variation as, just variation that exists in a population. Like, you wouldn't expect humans to be uniform, right? So why do we expect Boletus edulis to be uniform? It's okay to have variation within our species concepts. So I think that's maybe sort of a trend that's beginning with taxonomy of mushrooms in certain groups. Hopefully, I answered something there. Okay. Who or how are decisions made to decide on names? Is there citizen science community consultation involved? Some forum. Yeah, that's a very interesting idea. So I think by and large, the citizen science community is not um, consulted by people who named species. Anyone can name a species, however. So it's not that naming is, only to be done by professional mycologists. In fact, I would say there's a really important role for citizen scientists in naming species, because a lot of professional scientists don't, they're not, the naming species is not valued by their employers. And so it's not something that a lot of us can um, justify spending a lot of time on. That said, I think there should be a balance, because if we have this huge proliferation of species suddenly that don't have a lot of rigorous evidence to support them, then I don't think that's serving anybody's interests. So I would love to see more discussion about species names between the people that name them and um, the wider community, both professional and the citizen science community. Um, and I guess that's my answer.
We have a couple questions over in the chat, if you could take a look at that too.
Okay, let's see. Okay, for those of us collecting boletes in North America and using standard texts and keys, can you comment on how we should think about your results affecting our field classification exercise? Oh, I didn't answer that. I kicked that one down the road. um, so I mean, this is tough. I'm going to kick it even further down the road and tell you that I don't know. I would love to see some kind of I mean, one of my dreams is to see like some kind of community consensus where everyone can agree on the genera of the Boletaceae. I don't know how realistic that is, to be honest. But I would like there to at least be a set of um, guidelines for naming new genera that we can agree on if we can, because I think that will serve everybody the best. Is if there's an agreement about what lines of evidence do we need to name a new genus, what do we think is important to preserve in those names, then I think the whole community will be served. So I have my own ideas, of course, but I'm sure plenty of people have other ideas. And um, I guess I'll give a plug now. I'm organizing, or I've organized, co-organized with Keaton Tremble, my former grad student, a workshop at the IMC, and we are hoping to broadcast this workshop live, and we're going to discuss Boletaceae taxonomy, and the main point of discussion there is how we name new genera. And what I'm hoping to do is be able to broadcast this on Zoom so that people who are unable to attend the meeting, as well as citizen scientists, well, especially citizen scientists, can participate in this discussion. I know it's not going to work for everybody because it's on a European time, in a European time zone, but this may be at least sort of a launching off point where we can broaden the discussion amongst a larger group of people. So that's me punting it down the road even further. Sorry. Has there been any attempt to put similarity indices to differentiate one taxon from another? For instance, taxa might be 95% similar to be conspecific. Yes. In fact, ecologists do this all the time. In the standard cutoff for fungi has been 97% similarity. But the problem with that is similarity. This is something I tell my students: similarity does not equal relatedness. And I'd have to go into a longer discussion, which I don't have time for tonight, to explain what I mean by that. But suffice it to say that you're going to be misled by similarity in some cases where it's important. Not all. Maybe similarity works in most cases, but sometimes the devil's in the details there. So, and then should we consider differentiating morphospecies from genospecies? Yeah, I mean, this is a topic. This is a very broad topic about species concepts, and I clearly don't have time to get into that. But this has been a topic of conversation for many, many decades, and there's not a single solution to this. So yeah, I'm gonna have to leave it at that. A few million years. I mean, that's my preference, is to keep it simple and just call it one thing for now. Okay, on the colored bar graphs, why do some of the crossover genes show on the top and some on the bottom? Colored bar graphs, sorry, hold on. Are you talking about these?
Yes.
Okay. And why do some of the crossover genes? Oh, I see. They're just ordered. It's just the order. Yeah, that the being on the top or the bottom doesn't matter. I don't, to be honest, I don't know why Keaton ordered them the way he did. Um, okay. Do you have any measurement of how old is the gene mixing among Boletus edulis? We do, actually. We think we have very precise measures, and that's right here in this figure. So we have dated with a fairly high level of certainty that Boletus edulis is approximately 2.7 million years old. These red lines represent our confidence in that number. So you can see that it's plus or minus a bit, but not so bad actually. This is pretty tight for a confidence interval. So we're pretty sure that these have all been in the last 3 million years, let's say. And then the mixing, I think, how old is the gene mixing? So the gene mixing goes back all the way from the beginning, but we've been able to detect it in the recent, and we've done some modeling on this. And it's too, I don't have time to get into it, but I refer you to this paper where we do discuss the modeling of that if you're interested in reading about it. Okay. Hi, Roy. Taxonomic interpretation is a hypothesis. Hypotheses are tested, then have an unsupported lack thereof. Hypothesis can be debunked, supported. Exactly. Yep. Exactly. Yeah, and I think you raised a really good point that I didn't mention, which is there's one truth in taxonomy, and that is always changing, right? Because we get new evidence, and that's science. That's okay. Okay. It seems from what you presented that protein coding genes didn't give consistent results in identifying genera. So what can we use to separate genera? I feel we don't know enough about life histories of mycorrhizal fungi. Yeah, so all the genes that we use in our phylogenomics, so the genomic genes, those are all protein coding genes. So I think the thing to appreciate here is that there's not any one gene that's going to give you the correct signal. That's why we need lots of genes, because together we can infer the common signal. Hopefully that explains it. Okay, and then back in the Q&A, I'd like to know more about the undescribed species in North America, especially about Texas. Yeah, so me too. I can tell you that these are all posted on Mushroom Observer. That's how we originally found out about them. So I would let's see, I would recommend going to Mushroom Observer, or maybe I'm trying to think of a more efficient way than you just trying to find it. But I believe, so actually, if you look at the study that we just published, there's a Mushroom Observer number associated with the specimen in the supplementary information. So you'd be able to find that in there and go to it directly. We need more samples, I should say, to be able to describe it, because I think right now we only have 1 or 2 specimens, and I'm sure there are more out there. And we need more to be able to sort of uh, get a handle on the variation to be able to describe that as a new species. Okay. Is gene mixing being affected by distributions caused by humans? I love that question. Yes, it is. And there's a great example of it right here in the map. You'll notice that there's a yellow pie chart in the middle of Eastern North America. Well, that's an import from Europe, we're pretty sure. So that sample the genotype is almost 100% European genotype. And it occurs on it occurred near planted European trees. However, so we think that is a case of an import that is probably contributing to gene flow in this region. But I will also say that there's a lot of European trees that have been planted that have been colonized by the native genotypes. Send samples. So I would say get in touch with me through email. I'm sure you can find it if you just search my name or go to dentingerlab.org. My email should be there. I should get on to Mushroom Observer. I'm just yeah. So there's not 100% overlap between Mushroom Observer and INAT. Unfortunately, I end up sort of soliciting material from both sites because I know that there's not 100% overlap. Citizen mad scientists digging up edgeless hosts on different continents and planting them in the other regions. Yeah, right. Yeah. I will say that so far, and then I'm gonna have to close this out. But so far, all of the genotypes that we've seen from the introductions to South America, Africa, Australia are all European. I'm typing my email in the chat. Please be patient with me if I don't respond to you immediately. I get a lot of emails and I'm not always the best at responding immediately. So also, like, please follow up if you don't get an email response from me in the time period you wanted it. Trent can tell you how difficult it is to communicate with me, right, Trent? Just please be persistent, because it's not that I don't want to respond. It's just I have a lot of things going on and I'm not the most organized person. Okay. All right, I think I'm gonna end it here, because I gotta go. And yeah, thanks for sticking around, everybody.
So much, Bryn. That was great. I learned a lot. My brain is stuck right now. So I took a couple screenshots, and we're going to put this up on YouTube for anybody that wants to watch it again and learn again. Thank you.
Cool. Great.
Good night, everyone.
All right. Good night, everybody. See you again soon, I hope.